Good afternoon. Happy Friday. Everyone ready for the weekend? Yes. Okay, so today it's a lecture divided into two sections. One is pharmacology and medchem. I will cover pharmacology and Dr. Chatterjee will cover the medchem portion of. We have two topics of discussion here. One is topoisomerase and the other one is microfeedback. Okay, so drugs related to those two, we are going to talk about. Can anyone hear me, or do you want me to use a mic?
The reason I'm saying, like, while you are recording, it won't catch it.
Oh, it won't catch it?
If
Okay.
you are away, like you're going away,
Okay.
if you're near here, it's fine.
Oh, okay, okay. Let's then get that. Because if we want to get a good recording, yeah. The only thing is with the, I will have to hold it.
No, you can put it in your pocket. You don't.
Oh, yeah, yeah. I do that. Just, hello, is it better? Okay. So let's begin. So let's first focus on the topoisomerase. Sorry. I don't know what's happening. No?
That is not turned on. The remote. The remote has been turned on.
Yeah.
On off button.
Okay. I see. Yeah. Technological glitches. Let's go figuring out. So here are our learning objectives as the whole, but the blue one is the medchem learning objective that Dr. Chatterjee will cover. And the other are pharmacology learning objective for topoisomerase topic. So let's first review a little bit of topoisomerase because I'm sure this was covered in biochemistry. And Dr. Chatterjee have already covered a lot of it in detail about this enzyme. But do you guys know what topoisomerase is?
We talked about topoisomerase. Don't say like.
That's cell function, right? Topoisomerase is where does it exist? First,
Transcription, translation, and finally translation, right?
Yeah. So it's important, right? But this enzyme is going to be in which part of the cell? Nucleus. I heard nucleus, right? So let's see, before we go into that enzyme, why are we interested in this? So again, as Dr. Chatterjee said, this DNA replication and DNA synthesis, we have been talking about that in biochemistry, but everyone knows DNA, right? So DNA, how many of you have extracted DNA and run DNA on the job? Anyone? Yeah, maybe few hands. So you know, like, is it single-stranded? Is it like what kind of DNA do you visualize in your head? Double-stranded DNA, but is it like a thread or like what kind of? It's helical and it's like really complex. Like it's not a single thread. Like it's coiled, right? It's very coiled. So visualize like a ball of wood, right? So the DNA needs to be condensed. So think about nucleus. Is it visible to our eyes? The nucleus? No, right? You have to look at it with the microscope. And if the DNA has to reside in the nucleus, can it reside like a floating, like single molecule, or does it need to be condensed, right? It needs to be condensed into a small ball, right? So those are really very small structure. DNA. It may like in a book, it may look like a ladder and double-stranded ladder, but actually in real life, in a nucleus, it's like a really, really tiny condensed bond. And how do you condense that much? The two different structures are responsible for condensing into small bond. One is supercoiling, which is as represented here, and the other one is knots. Means not only just supercoils, but it creates knots to condense further. Supercoiling is one important step, and then also is knotting. So don't worry about all the details on this slide. What I'm asking you to pay attention is this is one structure very critical, and for today's lecture, and then the other one is knots, and the knots are created in two different ways. Like the DNA recombine, so they recombine, it has the threads pass through and causes knots. The other one is circularization, so the DNA gets circularized, and that also creates knots. So these are just for you to appreciate. It's not like I'm going to ask you what is knot and what is supercoiling. It's just to ask to understand what those two terminology means. When I say knots and when I say supercoil, these are two terminology you need to understand what that. Okay, so now that we know what the DNA, how it exists, let's go back to what is this enzyme, topoisomerase, what topoisomerase is doing. So generally, like as we mentioned, it's in replication, transcription, also in DNA repair. This enzyme is really critical to perform those functions, okay? So how does this do? It cleaves and reseals the phosphodiester bond, which is existing in your DNA at the backbone. So that will help in unwinding of the DNA and relieve the stress. Because if it is so coiled, supercoiled and not, then you must wonder, like, how is transcription taking place when it's so tightly condensed? So the body has, in our evolutionary way, it has built these mechanisms to unwind or do supercoiling. So it goes back and forth. It doesn't remain in the same stage forever. If it is some part may be supercoiled, some part may be uncoiled, depending on where the gene needs to express and what the cell needs to do. Okay? So sometimes we get caught between these terminology, but we don't try to visualize. Try to visualize DNA. And then you will be able to appreciate these functions very clearly. Because if I'm thinking like everyone has seen, these tangles and knots in your life in different ways, like ropes, and sometimes your chains get tangled, in jewelry. So there are different things happen around us which we remind us of tangles and knots. But in DNA also exists those tangles and knots and they're breaking that. So going back to that, like here I'm just showing you how this double-stranded DNA and the enzyme, don't worry about the details. So what I'm asking you to notice here is see how it is clipping and removing the stress and uncoiling that part of the DNA so that the other cofactors, everything can go and join and initiate transcription. I'm not going to talk about transcription today, that's not my intent. What I'm asking you is pay attention to the role of the enzyme, what is it doing, okay? So the another one, this one is supercoiling. When I'm talking about knots is where this is how the DNA is further condensed to form those knots. So these are kind of knots. So it's also relieving that. So there are two different types of enzyme. In a bit, we'll explain how that happens. So here, let's first think about topoisomerase 1. Okay, so think about topoisomerase as in general is like a scissor. When we have these tangled strings at home, how do we want to get rid of those tangles? Sometimes we cut it, right? So that now if you have a string, threads open up. Or if there is a knot, you try to loosen up, right? And then pull through the thread so that we can remove the knots. So think about these enzymes is your scissors at home. The good thing about these enzymes is they are not just scissors, but they also can repair. But our scissors at home would repair, right? Once you cut the thread, it's cut. But here, they can repair. So that's the beauty of these enzymes. So these are the scissors of our body. And pay attention to that. We'll come back to that thought process again. So here, so the way it works is here, look at this double-stranded DNA. And the way I'm going to explain to my slide is just bring you like a story, talk about, but then all the details are on this slide. So you can review later, and if you have questions, please ask. So this DNA gets attached to this topoisomerase 1, and it will clip. As you can see here, the pair of scissors is like clipping, right? It clips that and then relieves it. So this nick is going to let it pass the other strand through the cut, and then it reseals. So it's not just cutting, right? It's repairing by resealing it. So that's the process these enzymes can do very well. So the topoisomerase 1 can only cut single strand. So it's not cutting both the strands, it's only cutting single and then relieving the stress. And this whole process does not require ATP. So it's ATP independent. So if I ask you, does this enzyme, how does this mechanism work? Does it require ATP or not? We need to know that, okay? So the second one, as I mentioned, there is another topoisomerase, which is topoisomerase 2. And this one is causing double strand break in the DNA. Because it's hard, think about it, it's too condensed. It's hard to just only make one cut and relieve the tension, right? Because it's too supercoiled, too condensed. So those, you can see in this DNA here, this is how the melting happens, it's supercoiling plus. So supercoiling is here, right? Supercoiling, but then it's also condensed by putting these knots. So then this particular enzyme is cutting, see, you can see both the strands are cut. And this particular mechanism needs ATP for this process to happen. It requires ATP, it requires energy. So body needs to provide a specific reason why a condensed DNA needs to be loosened up. It doesn't want to just do it for no reason, right? So this particular step is critical, and that's why you need ATP. And once you have this, then it again that gets relieved and opens up, and then it relieves the condensation. So it gets decatenated. Okay, so it's more relaxed. Don't worry about this. It can get condensed again. So it all depends where your DNA, what your DNA is needing. So it's a reversible process, you know, so the condensation and decondensation happen all the time. So the topoisomerase 2 is for this process, for notch removal. Basically, one is for notch removal, the other one is for removal of the blood stress by unquote. So here is a summary of what I've spoken. So topoisomerase, one more thing I want to mention, it's a monomer. Mono means single, right? So it's a single form. And topoisomerase is a heterodimer. So there are two different, it forms in a dimer, these two molecules are together. And hetero means they are not the same. They are two different, but they are part of the same molecule, topoisomerase 2. So here, I already said it's a single strand break and religation happens. Here it is a double strand break and religation. Here it's going to relax the supercoiled DNA, and by relieving tension, here it relaxes DNA supercoiling, but also fixes knots. Also condensed DNA, supercoiled DNA. Does not require ATP, it requires ATP. Okay? So this is the table. Please pay attention to the differences, the similarity and the differences. So, okay, coming back to that thought process about scissors. So if the scissors are available all the time, then you will be keep cutting and pulling at random times, right? But in our body, we don't want that to happen. We want on very specific times and very specific places and during cell development where this process is needed. So these scissors, the topoisomerase levels changes. Their levels changes, their activity changes. And when? During cell cycle. The cells are going through this. Everyone, I think, through the biochemistry class, everyone has reviewed these concepts, right? The cell in the G0 phase, what is happening in G0 phase of the cell? So G0, zero means nothing, right? So they are just resting. So nothing is happening, like they're doing, in a growth wise, there's nothing is happening. But physiologically, that cell is still doing something, but it's not growing. So if it remains in G0, you don't have to worry about replication. You don't have to worry about that, right? Transcription replication of that. Cell dividing, going into mitosis, you don't have to worry about. But then as soon as the cell enters from G0 into the cell growth phase, which is like G1 phase, now it enters the cell cycle and then the whole process is going to get completed. I'm not going to get into every detail of that process because that you already have learned, and I'm not going to test you on that either, because you have been tested in biochemistry. So what I want you to appreciate here is let's first review quickly what is happening. So G1 cell growth, S phase, there is DNA replication, G2, there is preparation for mitosis, and then the cell division is occurring in the M phase, which is prophase, metaphase, anaphase, telophase, cytophases. Okay, pay attention to what is the major role here. In G1, there is cell growth. S phase, there is DNA replication. G2, there is mitosis, preparation, and M phase is the cell division, and then keeps going. It can go on and on and on. Divide from one cell to two daughter cells, from two daughter cells, and on, right? Until the cell exit the cell cycle. They can enter and exit, both. Okay, so here, these topoisomerase levels are changing based on what topoisomerase we are talking about, and their activity can also change. So that is what I want you to pay attention to, okay? So here, what in this table is showing you here is different cell cycle. So let's review topo 1. So topo 1 level actually does not change significantly during cell cycle. So it remains kind of very similar. Level, I'm talking about levels. But some studies have shown that their activity actually increases significantly during S phase. Okay, so they are there all the time, but they're really active during S phase. So active means they will start relieving those tensions. Okay, and single strand break. So let's now review topoisomerase 2. So topoisomerase 2, in G0 and G1, the level are pretty low. Okay, so this is what it means when I have this green, the activity of topo 1 is high in S phase. Okay, whereas in the topo 2, it's like you can see majority of the places where it's important for mitosis, both during replication, during preparation for mitosis, and during cell division, its activity is really high. So those two distinctions you need to understand. When the question comes like how your drug is acting, this information is important to analyze. Okay, so any questions so far before we move to the next topic? So now you have gained the background about DNA, what the DNA looks like, how it's, and then now you also saw how this enzyme is critical. Why are we doing this? Why are we paying attention to this? Why are we interested in this? Why are we using this enzyme for today's lecture? Anyone?
You want to change the part that is that asks that. So when you have cancer, it's that your cell is growing more than it should be because you have a problem with DNA, so this drug basically breaks a symbol that
Kind of, you're going in the right direction, but what exactly in cancer cell, what we will see today is one thing we all know in cancer cells are dividing a lot, right? Uncontrollably they are dividing, right? And that is what we are trying to control here, division. And how would you control division? We just saw that the cells are progressing from here to here to here to here, and then dividing again back and all over again, right? What is happening is topoisomerase is always acting and dividing, helping divide these cells. So that is why we are interested in this particular enzyme, because we will, if the scissors keep cutting, what do you want to do? Remove that scissor or block that or make it blunt so that it cannot cut anymore, right? So that is why we are trying to address topoisomerase. So here, in the cancer cells, so what is happening, let's review first. So, yeah, so here is your DNA, okay? And here is your topoisomerase, which is clipping. But if you provide a drug, so cancer drug, which we will talk a little later, just think in general, that drug will come and attach to this topoisomerase. If that drug is already attached to this topoisomerase can no longer now act in the process of replication. Okay, so that will get blocked and that will perturb the whole replication system, leading to cell death. Okay, so here, main thing we are doing is trapping. We're trapping these scissors and putting kind of like a block. So instead of like now we cannot cut anymore. So yeah.
Are there different enzymes that target the both topoisomerase or just one drug target the both?
So are there specific drugs for one or the other? There are some, which we will talk about in the, you are thinking on it, which is good. So we'll review those specific ones. But that's a good question. We will review in a minute, okay? So first we are talking about the whole mechanism at this point, irrespective of which topoisomerase drug we are targeting, but the main mechanism is going to trap it, okay? So that drug will go and trap, and then cell death will happen, okay? And the other thing I also want to mention, not only the drug, but there are mutations associated with these enzymes or within the DNA, like mutagenesis, or that's leading to mismatch, mutation mix, or recombination can also lead to cell death. But that's a concept I wanted to, put out for you. Not only the drug, but the DNA has its own way of mutating and controlling uncontrollable cell growth, okay? So not in the cancer patient, I'm talking about in general in the physiology, that sometimes the mutation and recombination can also lead to cell death, okay? And that can also trap so this enzyme can still get trapped because of that. Not only the drugs, but mutations and these alterations can trap that. So this is a question for you as a learning objective, like review it. We don't have time today to go through this question, but please reach out to me if you still don't know, do not understand, or are confused about what is the right answer. We can review later if we have time, but today we have to cover a lot of topics, so I will skip this. Oh, you didn't see that, right? Okay. So topoisomerase inhibitors. So now let's talk about the drugs. As we learned about the enzyme, the mechanism, how the drugs are in general going to attack this problem in cancer cells. So let's now be specific about what these inhibitors are, which enzyme, which inhibitors are specific for one or which are specific for two. So again, going back to our cell cycle, I already mentioned to you. So basically, the whole thing about these inhibitors are we are trying to control the cell cycle, right? Because cells are dividing uncontrollably. So our ultimate goal is to control the cell cycle. So these inhibitors, guess what? What are they going to inhibit? Topoisomerase 2 or 1, right? So that what will happen, the impact on the cell would be this now, all this enzyme is doing is nicking, right? Cutting and resealing. So the cells can divide again. So what we are doing is they can still cut, but they cannot be repaired. So they remain unsealed, okay? So that causes a disruption and leads to replication error, and then the chromosome separation won't happen. Because during mitosis, the chromosome needs to separate and each has to go to daughter cells, right? So by not sealing is able to do this, like replication error and no chromosome separation, chaotic division or leading to cell death. Because if it's chaotic, means the cells are trying to divide, but it doesn't have the proper separation of chromosome, it will cause cell apoptosis. So basically, yeah, this particular inhibitor is going to block these two. So now let's take a look at a drug class called camptothecins. So pay attention to what all these reds are, okay, in the slide, because that will guide you what is the differences, what is the mechanism. So this is in detail. The two things I'm going to explain is one is mechanism of action, the cell death pathway, how is cell death achieved, and what is the impact of all this. The impact, let's review, is primarily targeted to cause cells to, in the S phase is where it's going to happen, okay? So let's first see in the mechanism of action exactly how that happens. So this drug class, which has many different drugs, all of them will have the same mechanism. So it goes and traps topoisomerase 1. So this is DNA, the supercoiled, you can see, right, double strand. And then you can see these are the knots. So it's not attacking any of these knots. What it's attacking is the supercoiled trap is trapping the topoisomerase 1 here. The topoisomerase 1 was supposed to cause one nick, right? Single strand nick. So that once it's going to nick it, it's not preventing that. What it's doing is it's trapping them there so that it doesn't seal that nick. So it goes and does that, and then it produces, because of that, it's stuck, leads to double strand break, and that is irreversible. That cannot be fixed, cannot be resealed in any way or form. So that double strand break is the key here. So trapping and double strand break leads to the problem, targeting the cells which are mostly in the S phase. And makes sense, right? What did I say about topoisomerase 1? In that previous slide where I had the green and the red thing. It's targeting the S phase because of what? Where is topoisomerase 1 activity highest? At the S phase, right? Its levels are the same everywhere, but the activity is highest at the S phase. That's why it's targeting the S phase of the cells. So now it cannot replicate any further, so that's when all the replication machinery is encountering a problem leading to DNA damage and the cell suicide. So now it's a good thing, right? The cancer cells are trapped now. They're not going to go into this phase of cell cycle. So it significantly killing these cells. Okay. So now we have the second class of drug, which is the, I'm not going to say it's apidophyllotoxins. It's like a really big name. It's There are many drugs within that. All in this one, it does is it traps topoisomerase 2 instead of topoisomerase 1. This drug will trap topoisomerase 2 and then causes the single and double strand breaks. And again, goes to that irreversible DNA breaks, it cannot repair. But in this case, is going to topoisomerase 2 is active in S, G2, and M phase, right? So it will cause the arrest here. All the cells will not. So its activity basically is targeting the cells in these phase. So S, G2, M phase. And so at then once the transition doesn't happen from the S phase to the M phase. Okay, so that's where how it is going to do. So the third class is a little different than these two classes that we just spoke. This is anthracycline. So anthracycline is an antibiotic. Okay, so these antibiotic and all these biochem, medchem properties of these drugs will be this Dr. Chatterjee are going to elaborate on. So here it has very high affinity for DNA. So you can see red. Red is because what is rubicin? That word means to you. Ruby. What is ruby? Red, right? So this drug has a red color. If you put it in cells, the cells have red background. So that's why. So it intercalates into the DNA. You can see the double helix. It goes and intercalates it. And then stabilizes again the topoisomerase. So this complex gets, bundled with this DNA complex. So then there is no resealing again, replication is inhibited. So this has a little different way of killing. So it's disrupting the DNA replication by intercalating your drug within the DNA. So it has no cell cycle specificity because it's not an enzyme, it's targeting the DNA, right? So it will kill the cell cycle mostly in the S phase, but it can have effect in any phase of the cell cycle. Okay, so basically the cells won't divide anymore because the DNA is trapped, the enzyme this whole complex is not going to be working now. So it comes with a secondary effect, which is like cardiotoxicity because of this particular complex leads to free radicals which are damaging to the cells. So that causes, in some patients, not every patient, some patients have, based on their genomics, they have cardiotoxicity effect because of this. Okay, so the last but not the least, this particular drug class, mitose and santron, is very similar to doxorubicin. So I'm not going to explain again. It's the same mechanism. There's no red color associated with this, but this drug also intercalates into the DNA and traps your topoisomerase 1 and also topoisomerase 2, and then the replication is inhibited. So again, so any questions so far on this? If you have any, please come by when we have a little break. But let's finish the medchem portion of Dr. Chatterjee will preaching.
So, as Dr. Joshi Mukherjee explained to you in detail the mechanism of action, I'm going to make sure you are bored to death with all the structures. So it was the fun part which happened already. Now all the structures, yes? That's fun for me, right? Okay. So the thing, what we will talk about are the classes of drugs Dr. Joshi Mukherjee already mentioned, and we will talk about their characteristics. Primarily, we will focus on the pharmacophoric properties, because that is the thing what determines how the drugs are going to be biologically active, right? The pharmacophores. In going to that, the first slide, I would say that this is one of the slides you should pay attention quite a bit. It is a summary slide of all the drug names, so it is helpful. I did not bring my iPad, so I cannot put asterisk. But if you would like, you can put asterisk in this, okay? So these are the things what we are going to talk about today. Now, I try to make it a little bit colorful, so at least even though it's all structures, it really catches your eyes, right? And get some particular attention with the color coding. So primarily what we are talking about here, the first drug class, camptothecin, of which topotecan and irinotecan are the drugs. So they are sometimes called as camptothecans as well because of their last name tecan, okay? Now, these target primarily to topoisomerase 1. They are topoisomerase 1 inhibitor. Dr. Joshi Mukherjee has pointed out very vividly what is their course of action, right? So why we are even think of dealing with this topoisomerase 1 inhibitor? Because primarily our target is to stop cell growth of fast dividing cancer cells, right? Now, if we are thinking of these drugs killing fast dividing cancer cells, like in any other chemotherapeutic agents, they are non-selective, right? They will target the human fast dividing cells too. So it is a mechanism through which we are trying to achieve the best outcome possible to like with the given solution to us, that we are trying to kill the fast dividing cancer cells. That's our primary target. No cell division, no mitosis. That's the target. What we are doing, we are attacking either the topoisomerase 1 or topoisomerase 2, stopping replication. So it will stop the cell division eventually. Now, coming back to what I wanted to say, what do we see in these molecules? These are really, really cumbersome big molecules, right? What do we have in here? We have a ring structure, which I identified separately. It called camptothecin. That's essential. Intact. Camptothecin is essential constituent of all these drugs, which is the irinotecan and topotecan. That's the first required pharmacophoric feature. If we move on, what else? Free C10. But we don't see any free C10 oxygen, right? In here, particularly this irinotecan, we do not see free oxygen in the 10 position. That means no phenolic hydroxy in the 10 position is open for activity. So what do you term this compound? Is it a prodrug or is it a regular drug, like ideal drug? In circumstances, can this be generated into the active molecule? That's my question was. Can you think of if O is protected with this group? By the way, this group is a carbamate. If CO, nitrogen, that's a carbamate group, carbamate. So it is like an ester, right? So if you have, instead of ester, you have carbamate, that means amide variation, right? Then you have some kind of protection. So you can cleave that open in situ, right? In vivo, in the body. So this is a prodrug. which can be generated to the active drug in situ by cleaving with esterase, right? So it can generate the necessary fundamental reason for the activity of these two molecules, this free phenol. That has to be must. It is plant alkaloid. I talked about alkaloid at the beginning of biochemistry, the day one. Any idea what are alkaloids? Any definition of alkaloid? These are plant derived. I am already putting it in there. But the primary concept of this plant derived, they has to have nitrogen in there. Okay? So these are alkaloids. And the other thing is, in this compound, as well as in this compound, what do we see? Extra characteristics, and I highlighted with the color coding. Extra characteristics, what we see is the positively charged nitrogen compound. That means it's making a salt, right? It has a tertiary amine, which is made salt with a quaternary salt, right? Nitrogen makes quaternary salt with the anion floating in there. So what does it do? If you have an ionic compound, it helps in solubilization. So water solubility increases. So these drugs are water soluble, can be given in IV. So we already talked about the 4 pharmacophoric features, of which the 4th one is not kind of pharmacophoric, but we can improve the property by putting a salt. That was the thing. So coming back to what I just said, the 10-carbamate of irinotecan must be hydrolyzed in the liver to liberate the essential O10 phenol, makes it a longer half-life compound. We talked about that in ADME, we talked about that in biochemistry. A drug which is given as a prodrug has longer duration of action, because first it has to be cleaved, then the action comes into play, the active molecule, right? So it takes longer, 2-step process. It's not like something is already available to act. Whereas the topotecan, which already have the free phenolic group, phenolic OH, its half-life is short. It's very much self-descriptory, like already it is described as a self-description. Like the subject matter already states by itself what it says. Now, both camptothecans, what do we see? They have a lactam like that, right? Both of them, the E ring is a lactam. Yes? So, in the pH 7.4, which is slightly acidic, this lactam, or lactone actually, this lactone, it's not the lactam. Lactam is when the nitrogen is there. It's the lactone. This lactone gets hydrolyzed. If it does, that means it's a metabolite, right? This metabolite is a weak metabolite, inactive metabolite. So, in turn, we need to have activity persistent in the molecule, the drug molecule, if we have uncleaved E ring. That means in P453, 450, with like CYP450 enzymes, they are going to cleave this lactone to this open form. That's not good. Once it does, it deactivates. It deactivates the metabolite, what it forms, it deactivated. That's the primary thing. Now, irinotecan is a piperidine hydroxylation. These are the pathways where it can happen, the metabolism. Amide hydrolysis and O10 glucuronidation. Do we know, do we recall anything? Because in now on, your life is going to be full with description from the ADME concept, what I taught you before in the first year. In all the PT courses, you will be bombarded with all the ADME concept. That means CYP enzymes, which is which are responsible for cleaving it and whatnot. So if we recall, first pass metabolism are the regular reactions like hydroxylation, N-demethylation, things like that. But in the second pass metabolism, once you have this N-demethylation or O-demethylation, that means any hydroxy group open, they are susceptible to form sugar derivative. That means glucuronic acid derivative. Why? So that they are easily soluble in water and get out of the system of our body. Excretion happens. So this glucuronidation is a second pass metabolism, phase 2 metabolism, where oxygen like hydroxy, NH2, free NH2, they are glucuronidated with sugar molecule. That is one of the major pathway for both of the drugs. Now moving on to the next classes of drug. This is topoisomerase 2 inhibitors. For your sake, I made these as topo 1 and topo 2 so that with the slide itself, not only with Dr. Joshi Mukherjee's slide, but here also you remember which drugs are acting on which enzymes and so on and so forth. So these are AP photophytotoxin drugs. If we remember at all from the days of biochemistry, when we were talking about Taxol, I was talking about photophytotoxin 2. So these are actually photophytotoxin drugs. These are kind of arresting the cell division as well, but through a different mechanism. This targets topoisomerase 2. What does it have? Intact rings and functional groups as it is presented has to be must to keep its activity. That means it has a four-membered ring of this. The last ring here, anybody can recall what is this? What is this? This is an acetal. If we clip this, it will form 2CHO. So if you make acetal, that means from the acetal, like aldehyde, if you form a ring structure, you will form an acetal. This is easily cleaved through esterase again. So these are acetal compounds. At this position, the drugs are very soft. That means metabolically labile at this position. Other than that, what else we are seeing? We are seeing another cyclic structure, that means sugar molecule in there, disaccharides in there. And in the disaccharides, there are certain differences. These two first compound, etoposide and etoposide phosphate, they have just the methyl substitution, whereas in here you have a thiophene. So from methyl to thiophene, would it increase lipophilicity? My question is that if you introduce more aromatic character, that means more of the electron density in there, would it increase the lipophilicity of the molecule? Is it? It is actually so. It is going to increase the lipophilicity because you are increasing lipophilic bulk. So comparing these two compounds to this is more lipophilic. Between these two compounds, the only difference is this is a phosphate salt of this first one, etoposide. So if I make a salt once again with the theory of making salt, why do we make salt? To increase the solubility. So we are again trying to increase the solubility. This is more soluble than the free form. This is again plant alkaloid binds only to enzyme, not to DNA. These only differ at the C2 beta D gluconopyronosyl moiety. That means this is the disaccharide. Again, I said the difference is only between selecting either this small lipophilic group, which is methyl, versus the hyophene. Now, metabolism. Epiphytophyllotoxin must be solubilized for IV administration. All these compounds are extremely difficult to solubilize. So what do? They use some sort of surfactant. Again, if we recall the concept of lipid bilayer, we know that we can make things soluble by adding surfactant. So adding surfactant makes them soluble, as if the explanation or example I gave during the biochemistry days, that you have oil and water. In that oil and water, what you have, if you add some soap, it takes the oil out of that. Like if you have a cloth already oily stain, you add soap. That means it increases the surface tension. Same function for these surfactants, polyfer or twin. These are these has to be added to the compound to make them soluble. One of them, the first one, is only soluble in colifer, whereas the second one, the etoposide, is only soluble, can be soluble in twin. And it's been seen that twin is better compared to colifer. What happens because of that? Because of these surfactants to be used in order to solubilize these compounds, they cause hypersensitivity reaction. So even though you are applying that for the patients, it gave them hypersensitivity. Once again, coming back to the one, we talked about that to increase the solubility, we introduced a phosphate group. So this is phosphate group, makes it water soluble. Lactone hydrolysis, I already mentioned that. That's the soft corner, right? This is this lactone as well as this acetal. This lactone hydrolyzes to the hydroxy acid in a CYP3A4 mediated hydrolysis. That means phase 1, and that causes a inactive metabolite. So that's the whole target. As we are seeing for the other compounds, this has a lactone and it gets hydrolyzes. These are softer points. Now we are talking about the antibiotics. These are anthracyclines and anthracene diones. What does it have? It has really flat aromatic structure. As Dr. Joshi Mukherjee was pointing out, that they intercalate. Why do they intercalate? Because the bases, they are aromatic compounds. And we know, I showed you picture, molecular modeling picture before, that aromatic stack on aromatic, because flat stack on flat very well and thereby stabilizes. So intercalation, these are intercalating reagents. And they primarily target, they are topo 2 inhibitors in a sense, by intercalating, it does not let the topoisomerase 2 to perform. Mitosis stops. These are the compounds. And here you could see there are subtle differences in different substitution. I don't want you to pay that much attention to determine which compound is which, but general structure. What are the general structure? The pharmacophoric features. This ring, this anthracene dione ring, is essential. One. Second thing is, this is a dione structure, right? Middle one. This with reductase, again, phase 1, it goes to hydroquinone. If that is the case, along with forming the hydroquinone, it forms free radicals. That's really not good. It is toxic, cardiotoxic. Third one, this glycosidic bond is essential. If you have the breakage of this glycosidic bond, the compounds are not active. This sugar molecule in here sits, inserts this sugar moiety goes inside the group, thereby stabilizes the ternary structure. And the last one is this amine also is necessary for DNA binding. So here are the highlighted portion. I would say that these will explain what is necessary for the necessary pharmacophoric features for this molecule. Now, if we move to the last one, which is mitoxantrone, they are very much chemically similar to anthracycline. We can see instead of 4 ring, it has a 3 ring, but again, it gives a very, very flat structure, aromatic. So aromatic flat structure means it will sit on aromatic flat, thereby intercalate. It is planar, absolutely so, polycyclic structure, and cationic amino side chains are thought to bind to the anionic phosphate backbones. So this has some cationic nitrogen center. It helps in forming a chemical bonding. That means a polar bonding with the phosphate ring. Phosphate are negatively charged. These are positively charged. So it binds very well. Last thing I will say is methotrexate is the quinone oxygen. They are stabilized by hydrogen bonding. What does it do? So what do we need? Methotrexate's quinone hydrogen are stabilized by hydrogen bonding. What's the benefit of that? It avoids any CYP enzyme to come near it, so thereby protecting themselves. So this has longer half-life. The other thing is, once you have them already deaminated and carboxylated, the ultimate and only fate is to form glucuronide and getting eliminated from that. We have already seen glucuronide formation, which is a sugar attachment, making it water soluble to get eliminated faster. So in dealkylation, deamination, oxidation to the carboxy acid is the phase 1, and glucuronidation is the ultimate phase. That means the phase 2 metabolism. What did we learn so far? We learned are the pharmacophoric features of each of the drug molecules. You should be able to answer that. So basically hinting you towards what kind of questions you are going to expect. Identify the key metabolic pathways. I already pointed out kind of some phase 1, phase 2 metabolites, and identify the target sites, whether it's a topoisomerase 1, topoisomerase 2, or intercalating agent. That's all for this lecture. Take a break, take 5 minutes break, please come back so that we can get it done, okay?
We'll start with the microtubule. Okay, I guess we have to get started with the next topic. So again, in the previous one, we just talked about how we are targeting topoisomerase. Now we'll be targeting another, it's not an enzyme, this is another structural related protein. So microtubules are the one here we'll be targeting for cancer related therapy. Okay? So where does microtubule, where do they live in the cell? Like topoisomerase was nucleus, but what about microtubule? Cytoplasm. Cytoplasm, it provides a real nice support system, right? So microtubule, we'll review quickly the function. So these are the learning objectives for both medchem and also the pharmacology. The blue is medchem and the other ones are pharmacology. Okay, so
It was done now.
all right, did go. Okay, let's see.
Yeah, it should work.
Yeah, of course, of course. Let me just do one more time here. It's frozen here,
Frozen.
yeah,
You escape, like,
I did escape, but it's not escaping,
that's not good sign.
no. Because if we do Control Alt Del, then it will.
Okay,
All did?
Yeah.
Okay. So let's put it in the,
so you have to move it by hand. Move it by hand.
okay.
This is not functioning.
Okay, so let's,
Okay,
oh,
good.
it is function.
functioning.
Okay.
Good.
Sorry, this keeps breaking on me. So let's talk about just a few minutes about what microtubules are important for. So basically, microtubules are the scaffolds of a cell. So if you don't have a scaffold, the cell is going to die. So that is where we are attacking. We are removing that main scaffold of the cell. Okay. So here, what is scaffold important for? That scaffold is important for cell shape and structure. So which determines how the cell during morphogenesis and development, the cell shape changes, right? Like cardiomyocytes, when it's in a fetus or in a newborn, it's much smaller, more rounder, but as it becomes adult, it becomes like a raw shape cells. So that shape is determined by these microtubules. It defines those. Okay. So then this another important aspect it controls is the movement and the organization. So what does that mean? That during cell division, you need the chromosome to separate, right? Into different daughter cells. So that's where microtubules play a really important role. You'll see in a minute in the picture, you'll appreciate. And then in the transportation, makes sense, right? It's a scaffold, it's like a railway track where it takes all the goods, transports it. So it can transport organelles from one side to the other. It can also transport vesicles, which is in training, it's like goods train, right? So vesicles also is, it helps in transport. So you can see in this picture, appreciate how the cell, these are the microtubules, spindles, are how dynamic they are for the cell division. The cells are dividing from one to the other, so these are playing a big role where the chromosomes are, it's helping the chromosome to separate, okay? And another, I want you to pay attention here, this is a neuron. So this is just a television, this is the neuron, where you can see these tiny dots of, these are real. This is not just this is a real neuron, where the microtubules were labeled, and you can see they are transporting stuff from one corner of the cell to its neuron and dendritic ends. Okay, so this is such a dynamic structure that needs for all these functions. So when I mention that it's dynamic, what it means is it's constantly assembling and disassembling. In this, you can see right now it's adding stuff here, right? So it's assembling. Here in this tail end, it's disassembling. It's removing things. So these microtubules have many different accessory proteins. I'm not going to go into any of that detail. What I'm asking you to pay attention to is when it means dynamic means what? Growing, assembling, disassembling. Okay. So now, what it's made of. So I'm sure in biochemistry again you've been introduced to microtubules and what is it made of. Just briefly, it has two different heterodimers, so alpha tubulin and beta tubulin. So they are hetero, means they are different, right? Alpha and beta. So it's a dimer. So here you can see alpha tubulin has a very high affinity with beta tubulin, and it's GTP binding. So it's when it's GTP bound, it can hydrolyze to GDP, and then it changes this conformation and changes the property of this complex. So why we are talking about that in a minute, I'll show. So there's one more thing I think mentioned here, it's hydrolyzed. And this one goes through hydrolysis to form this, okay? And why do we need that? Because we are, again, when I said dynamic means adding things, adding to the head and removing things from the tail. So adding is the plus end of a microtubule is limited by this beta tubulin. And negative end is limited by alpha tubulin. So if you have problems with this particular protein, it's going to affect the positive end of the microtubule. If you have problems with the alpha tubulin, it will affect the negative end of the, yeah. And here you can see in this appreciated this is the microtubule. This is the microtubule structure. You can see multiple dimers of, so this color, the green color is represented here, this GTP bound tubulin, and the purple one is the GDP. It's already hydrolyzed, so it's here. So minus N enriched in alpha, and the positive N is rich with beta. Okay, and so once this is the assembly part. So plus N when it's enriched with the GTP beta tubulin, it's assembling, it's keep assembling, but then when there's hydrolysis, it starts disassembling. So it's a continuous back and forth process. So it can disassemble and rescue. Okay, so those are the two important concepts you need to understand. So what is the hydrolysis and how it's added, and so then it can, since it's dynamic, it can be unstable based on the hydrolysis of the GTP to GDP, right? So if this particular microtubule is here, is how much it's growing or reducing depends on the GDP to GDP hydrolysis. So based on the rate of hydrolysis, it will break down or grow. So if hydrolysis is faster than polymerization, then it's going to break down, means it will follow this process. But when the hydrolysis is slower than polymerization, then it's going to grow. Then it's going to be in this phase. So the two phase depends on the hydrolysis, basically the rate of hydrolysis, the rate of converting from GDP to GDP. Okay? With that information, everyone is with me on what microtubules are and how are they functioning. So let's now Why are we doing this? Because we want to target microtubules in these cancer cells, right? We want to kill them. We cause cell death in these. So we are going to remove these scaffolds by these drugs. So these are the class of drugs that we will be going to discuss this today. And all the medchem related aspects of these drugs will be covered by Dr. Chatterjee. But I will more cover about the mechanistic part, how are these drugs going to function, okay? So again, I said they're going to disrupt the dynamic behavior of microtubule. So this particular mechanism is kind of going to apply to all of your drug classes. So this is what first let's understand what that is. So the drug is going to form a complex with the tubulin, okay? And that complex will determine whether it's going to stabilize microtubule or destabilize microtubule. But both are disruptive, whether you are stabilizing too much or destabilizing too much of that microtubule is disruptive. And then that would cause cell cycle arrest. So basically, ultimate goal is to arrest the cell cycle. Mitotic arrest, this one, in phase. And how you're doing it? Because you're interfering with the mitotic spindle. Remember, the chromosome needs to separate for the cell division, right? So that's why you are trying to interfere with the spindle formation. And then that leads to inhibition of cell proliferation and leading to cell death. So these are the two basic mechanisms. So in that way, like if you want to visualize, you can see this is a continuous process in normal cell, but when you add a drug that will destabilize, this will move into this direction and then it will disintegrate the microtubule. Whereas a drug can maybe let it grow and grow and grow and cause bundle formation. So microtubules start bundling together, leading to they both are disruptive. Even having this or having too much bundle formation is also going to be detrimental to the cell, causing cell death. With that in mind, let's now review the drug classes. So this particular, since it's such a simple mechanism, I want you to pay attention to this table. I have divided these drug classes that Dr. Chatterjee is going to elaborate on the medchem part, but the mechanism, we can divide it into two groups. Very simple, right? I made your life simpler. In this microtubule, so let's first review the stabilizing agent. In the stabilizing agent is taxane and apotelones. These are two different classes, which will disrupt the microtubule dynamics by first forming a complex and enhances the polymerization, means is going to go in this direction. So the green arrow, pay attention to the color. So in the picture, this stabilization means this particular microtubule, when it encounters, it binds to this tubulin and forms a complex that will push this microtubule towards in this direction and it will start stabilizing. Means the microtubule will keep growing and form bundles. And that will lead to cell cycle arrest, okay? Whereas in this now we take care of the other destabilizing agent, which is halichondrin and vinca alkaloids. These two, now again it will form a complex, the drug tubulin complex, and that complex will add to the positive end of the microtubules to terminate the assembly. Means like now it will push this into this direction. So now these two things, the alpha and beta tubulin, because beta tubulin will bind to the complex, it will cap this on top of this, and this particular microtubule becomes unstable and disintegrates, destabilizes. Okay? So everyone understand the two different mechanisms? Any question on this? Okay, so now in this specific cell cycle arrest, what is happening is arresting from G2 to M cycle arrest. Means after, in the M phase, they will not proceed further. So they will get arrested either in G2M phase, and there is no division, okay? So that's the ultimate goal here. So another thing I also want to point out, and Dr. Chatterjee is going to elaborate on this. So then why do we have these two? I mean, we have these two classes, but what is the actual, each drug? Why are we discovering new drugs? Why are we, having so many different kinds? We can just pick one and treat the patient with one. Why do we need so many? So what I want to say is, in this beta, so this is beta tubulin and alpha tubulin, they have to bind to each other. They have very high affinity, but within this beta tubulin, they have different binding sites for these drug classes. So Vinca alkaloids has a different site, and helichondrins have different site, apothiones have different site. I mean, similar to taxane, but they have very high affinity. So epithelium is competitive binder, so it will bind, if they are there, they will bind this site better than taxane. So they are able to achieve different, by binding to these different sites, they are either better, have better target effect. So, and there could be other reason associated with more that is elaborated in MedCamp. So one thing that I wanted you to appreciate is that different binding site provides different specificity for these drugs. And also I want to show you where the GTP binding site is, and exchange both in the beta tubulin with respect to different binding for the drug. They are different. So they are not the same GTP binding sites are separate from these drug binding sites. So again, here just mentioning the vincristine has better affinity than this particular molecule than vinblastine. So that's all I think for the pharmacology. Oh, before I move, one thing that I wanted to mention is in all these drug treatment, we also see drug resistance, right? So not all the cells are killed. There are some still cells which escape the drug effect. So otherwise you can just treat it once and that should take care of the cancer, right? But we don't see that. There we see a lot of cells escape that drug effect. So there are some reasons behind that. So the mechanism is drug is binding that has a receptor. So the drug is taken up by your cells, right? So because we are saying tubulin targeted drugs, so the drug gets incorporated near your cells and it has to go in or it has to escape, depends, like that defines the concentration of the drug in your cells. So then what happens is there is overexpression of these pumps. When they are overexpressed, the more drug will efflux, right? So the more drug is thrown by the cells, that means it's not doing its job. So that is causing one reason for resistance, okay? So overexpression. So the next is the target, modification of the target. The drug is targeting what in this case? What are we targeting? Microtubules. So the microtubules are, if they get modified, how do we modify them? By mutation, and different isotopes of tubulin may exist, and then it can attach to a protein to the end of microtubule that the drugs cannot bind anymore. So those are the modification that's going to disrupt microtubule. So these are alterations. Basically, you are altering your tubulin and microtubulin. So by different gene expression, mutation, attachment to a protein. So this slide is really important too. So you need to understand why these resistance are happening. Also, there is post-translational modification. So post-translational modification of microtubule can also cause post-translational modification for phosphorylation, glycosylation, all those things, okay? And also reduce susceptibility to apoptosis. What does that mean? Means like you are, none of these are occurring. There is also, you are first arresting in mitosis, right? But if apoptosis doesn't happen, then you're not killing the cells. So you have to achieve that, means there is an anti-apoptotic signaling, means that the cells are arrested, but the apoptotic signal is not available. So there may be some anti-apoptotic factors are there to prevent it. Okay, that also can lead to the resistance. So what I also want to mention, this happens as a late event, right? First is drug accumulation. So the early is more like further drug comes in, and here down the path, even though everything happens, but apoptosis doesn't happen, then it's also causing resistance. The cell is not going to die anymore. So all this, if say suppose they are able to somehow repair the DNA damage, that can also cause resistance. And altered proliferation. For some reason, the proliferation is taking place in a different way than the way we are targeting here. So all of these reasons can be a cause of problem for not leading to cell death in these patients. So with that, I think, any question on the pharmacology? If not, then we can just move on to the next cam. Oh, before, please, sorry, this one quick thing. This is just a practice quiz. You can take it at home so that for the pharmacology concepts, I want you to take this quiz so that it will prepare you for your exam. Also to see where the gaps are, then you can come and talk to me and I can explain further, okay? So with that, finally, Dr. Sam, the stage is yours.
Thank you very much. So what I will do before going into MedChem, I just want to borrow a couple of slides from Dr. Joshi and try to see like at all whether you paid any attention during the biochemistry class or not, to make me feel a little bit good. In a sense, like majority of you probably did not pay attention. I'm just kidding, okay? And that makes me feel really good that, okay, you don't remember any of the concepts I try to teach, okay? Now, just going back one slide, I just want to check your knowledge, okay? Just revisiting. So Dr. Joshi was talking about drug efflux, yes? Which pump we are talking about? Any drug resistance, which pump is involved? Is it an active transport or passive transport? Go ahead. Henry? Active transport, right? Are we on that? What kind of pump? ABC transporter, right? Particularly Sarka pump, right? I said drug transport only happening through that. What is the transporter? Anybody remember? PGP, right? Phosphoglycoprotein, PGP. That's the transporter which actually carries things out. But in any way, it makes me really feel good that like at least Henry remembered, okay? And I'm going to borrow one more slide from Dr. Joshi. This is a wonderful, wonderful picture. The one I'm talking about on the top.
Central.
Oh, okay.
This one.
Okay. So this is the one I was talking about. What is happening during the mitosis? Means the telomeres are separating, right? Chromosome is separating, yes. So it is depolymerization is happening, yes, of the chromosome, like mitotic spindle, to form two daughter cells, and then polymerization, right? So basically, what I just wanted to mention in this mitotic spindle, where polymerization and depolymerization both works in a dynamic pattern, some of the drugs target depolymerization only, right? So it won't polymerize back, it depolymerizes. Whereas some of the drugs enhances polymerization. That means it keeps the drug polymerized. And I did mention that because it was very dear to my heart. In 1992, I actually published the first paper with Taxol. I synthesized Taxol, and that was an azido derivative. I was talking about the diazonium ion the other day, right? In the this alkylating agents, diazonium ion. So what does it do? Diazonium ion forms covalent bond with the protein. And thereby that compound was used to identify the binding site of Taxol. So I was very happy, and it was a very challenging synthesis. But in any way, forget about those. Coming back to what I was talking, that one is the mitotic spindle depolymerizer. Other one is mitotic spindle polymerization enhancer. In the depolymerizers, these two compounds leave, whereas in the polymerization stabilizer, these two compounds stay. As Dr. Joshi was pointing out, they have their own binding sites, and primarily in the beta tubulin, right? Alpha tubulin, where the hydrolysis reaction is taking place. Beta tubulin, where the drug binds with the to form a complex. Okay, so just wanted to point out that polymerization inhibitors, they also, like say for inhibitors like vinca alkaloids, they bind to a separate beta subunit compared to the polymerization, like depolymerization inhibitor. That means polymerization enhancer, they bind to a different subunit of the beta. Now, this is something we are talking about vinca alkaloids. Alkaloids essentially are very, very complex structure. Nature giving us compounds with very complex looking stereochemically active compounds. This is actually vinca alkaloids, means it's like collected from the vinca trees. The primary ones are this vincristin and vinblastin. Then there is another one which is vindolin. Vindolin, which is the vinorelbine tartrate. Tartrate is the salt. But without getting to the complexity of the name, all of them belong to a pharmacophoric class, which are termed as vinca alkaloids. What is the pharmacophoric features like important for this? The pharmacophoric features are, as you can see, for these vincristin and vinblastin, what they have hydroxy group compared to no hydroxy group here. Yes? So if we compare these two structures, which one will be more lipophilic? One on the left or one on the right? One on the right is more lipophilic because it is devoid of that hydroxy, which is giving some polar characteristics of the molecule. Then all of them has this vindoline portion, like basically this alkaloid portion, which is this guy. Yes? And what else does it have? This one, the N substitution, nitrogen substitution, is different for vincristine and vinblastine compared to this vindoline, like vinorelbine compound. What is the difference? In the vincristine, R is CHO. It's a formyl group. That means an aldehyde, which is polar. In the vinblastine, it is CH3. Whereas in vinorelbine, it is again NCH3. So comparatively, the one with the CHO is more potent compared to this CH3 substitution. So subtle differences between all these three. Let's see. This vincristine, because of this nitrogen substitution difference, vincristine is neurotoxic. Vinblastine and vinorelbine have neutropenia, whereas vinorelbine more lipid soluble due to differences in the alkyl chain. We can clearly understand. We may not be able to identify their toxic effect. That means the neurotoxicity or the neutropenia. But at least from the log P standpoint, we should be able to identify which one is more lipophilic and which one is less lipophilic. Just this portion, just you need to know that vincristine is neurotoxic. There are only 3 compounds, of which vincristine is neurotoxic, and the other 2 has to do with neutropenia. If you can, if it helps you in any way of remembering. Now we are talking about the vinca-alkaloid structure. This configuration, so primarily, as I was mentioning, that nature gave us compounds with extremely complex structure and with extremely difficult to synthesize compounds. Why? Because it's putting stereochemical integrity. That means it has some chiral center. Yes? The chiral center is something where you have 4 different groups. And we can easily determine, I'm not going to ask you that question, we can easily determine the stereochemistry of these 2 centers by assigning them R or S. And we know how to assign that, but I'm not going to ask you that question. So their stereospecificity is absolutely identified. In a sense, like they have particular stereochemistry in this chiral center, the chiral center here, and the chiral center here, which is specific, and the chiral center here. This requires this particular chiral arrangement. That means whether it's R or S, it is required for these vincaloid alkaloids to be active. If we have a different stereochemistry than what it is there, it is going to be inactive. So how do we remember that? Stereochemistry is in the vincaloid, you can write it down. Stereochemistry in the vincal alkaloid is the most important factor to keep its activity. Okay. Now we are talking to this R3 group substitution in here. This one, it is COCH3 or hydrogen, depending on what kind of, whether we are talking about vincristine or vinblastine. One is COCH3, that means a ketone. other one is an aldehyde, and we can easily identify which one is a ketone and which one is an aldehyde. Aldehyde is the one which is vincristine, whereas ketone is the one which is vinblastine. Now, in here are substitution, as we have seen before, that changes the nature of the compound in terms of neurotoxicity. One cause neurotoxic, the other one is neutropenia. So if you have a CHO, it causes neurotoxicity, whereas if you have a CH3 here, it causes neutropenia. I pointed that out before itself. Other things is this is a center where it can actually bind to the protein in different region. We can see that it can bind to the tryptophan, it can bind to alanine or valine at this position. So primarily, in terms of stereochemical, like in terms of SAR perspective, that means pharmacophoric features. What do we see? First thing we see is this vinca group. That means is this vindoline portion. Number one. You can even write it down because I have no way of highlighting this. So this vindoline portion, it is essential for activity. Other things, all the existing stereochemistry in the vinca alkaloid has to be present. If it is changed, it is going to be inactive. And depending on third point, depending on different substitution of this nitrogen, the toxicity changes. These are the three fundamental statements for this vinca alkaloid, like those pharmacophores or structural elements. We need to pay attention. Next one is helicondrin. As I have lined up, as well as Dr. Joshi has lined up for you, that we divided in two different classes. One is polymerization stabilizers, other one is depolymerizers. Now, at this point, I'm talking about depolymerizers because we know vinca alkaloids, they belong to the class of compounds which depolymerizes. Helicondrin is also a compound which helps in depolymerization. So either way, polymerization or polymerization stabilizer or depolymerization, both are similarly responsible for stopping the mitosis. That means cell division. Now, this is the structure of the helicondrin. Helicondrin B is primarily identified, again, nothing to, this is also a naturally identified compound, came from natural sources. What is that? It is a marine sponge where this helicondrin B is identified, like it's first isolated. What are the major, and then compound supply was a major obstacle to clinical development. Some synthetic modifications were done. Just by looking at all these natural products include compounds isolated from bark or tree. Those are called alkaloids, as well as natural products identified, isolated from sea resources, or like a spider venom, or like any other venom. So those are all natural products, and they are responsible for so many different biological purposes to help us cure some diseases. In here, we are dealing with compounds which are mitotic inhibitor. Here, we are talking about that this is, since the synthesis is very, very challenging, and there is limited resources through which we can get the compound isolated from marine sponge, scientists, drug discovery scientists have did some development to get simpler compounds made, which create the same or similar activity. What are the things got changed? First one is this guy. This C1 lactone is replaced by ketone. So this is the lactone here. You can see the lactone in here. Can we see the lactone in here? That is replaced by this ketone. Then this C31 methyl is replaced by a methoxy. Methyl is replaced by a methoxy, means there is little bit change in log P value. C29 to 38 is replaced by 5-membered ring. All this, basically this side chain is removed to get this really simple side chain version. And this small group of this whole ring system, which is a like 3-ring system, is replaced by 1-ring system. So basically what I was trying to point out in this halochondrin derivative, which is known as eribulin, is a simpler form of the natural product. Without to make your life more complicated with this complicated structure and the particular changes happening in the molecule, what I am asking you to do, if I give you a question in terms of what is the structural differences between the halochondrin and eribulin, a simple answer could be there are structural modifications done to the synthetic analog to access the same biological activity or similar biological activity, to make it much more simpler so that synthetically it is not that challenging to get the actual drug, which is the natural product. Moving further to, we are getting into the territory of polymer stabilizer, polymerization stabilizer. The first one to pay attention to is the taxane derivatives, of which Taxol and their cousins are there. Why I'm saying cousins? Because it does have the same Taxol structure, but some modifications were done to improve the bioavailability. How? To make it first more water soluble, so it can be applied without causing any side effect. What does it there? What is the primary thing? Primary thing is it has this ring is called phenyl isoserine side chain, this ring, right? And this is called baccatin 3 ring. This together is Taxol. Paclitaxel has this, docetaxel or taxotere, which is a much solubilizing compound or derivative that has this, and carboxystaxel has this kind of substitution. Again, I'm not focusing primarily on different variations of Taxols and what do they have. What I'm focusing on are the pharmacophoric features. What do they have? Functional groups that bind to the tubulin. Let's go to the Taxol ACR to see the pharmacophoric structure. What do we see? First thing, it needs to have 2 most important components. One is the phenyl isoserine side chain and baccatin 3. Those are the 2 most important components. Then coming back to the other thing, this benzoyl group is required in here. This acetate group defined the conformation of the molecule. Side chain, as I already said, that phenyl isoserine side chain is necessary for the activity. Basically, in taxotide, the difference is instead of acetate, you have a hydroxy, which gives it a little bit more water solubility. And in this baccatin 3 portion, what I want you to focus that it needs to have this oxy-10 ring. So highlight on oxy-10 if you can, or put a mark on oxy-10 ring, which is a 4-membered oxygen ring. So I already pointed out 4 important features. 1, baccatin 3 or taxane molecule. Taxane is that particular cyclic structure of the baccatin 3. Needs to be there. Second, phenyl isoserine side chain has to be there. It needs to have, this is most likely situation is the 13 and this is the 10. This 10 acetate group is converted to hydroxy to improve water solubility for taxotide, needs to have this oxy-10 ring. Those are the pharmacophoric features important for Taxol. Now moving on, this taxane medicinal chemistry aspect, as I was mentioning, this taxane looks like a big molecule and synthesis was extremely challenging. But in any way, coming back to what I was saying, this molecule looks like they don't have that much solubility. And as I was mentioning, this C10 position, I already specified. Please highlight that C10 acetyl group is replaced with a hydroxy group for taxotide to make it water soluble. Okay? But in any way, so what does it do? What is required for Taxol to be soluble? Again, you need to add certain surfactants. For taxotide, or that surfactants is not needed. So if you add surfactants, as I was mentioning before, it causes hypoallergic reaction, like certain reactions which is not ideal for patient's application. So you divide of adding the surfactants by increasing the water solubility for taxotide. That is all for the Taxol. The last class of compound, epithelons. By the way, epithelons also target, as Dr. Joshi Mukherjee was pointing out, it targets pretty much the taxane site where the taxane bonds, but it has more affinity. This is a much simpler structure. The compound is IXA, BPILONE, ixapilone. This compound, like paclitaxel or Taxol, this ixapilone requires solubilization with the polyphor-EL. That means it will cause some secondary effect also because it requires surfactants. And the amide linkage of ixapilone protects against the inactivating hydrolysis. Can we identify where we see the amide bond in this? Anybody? Is the amide in an acyclic position or is amide in a cyclic position? Is it a cyclic amide or just a linear amide? Anybody can identify? Yes? So it is a cyclic amide. We can see this cyclic amide, because of this presence, accessing, it protects against the inactivating hydrolysis. If you have a linear amide, there is question of amide hydrolysis. But if you have a cyclic amide, the CYP450 won't be able to target that. So that is primarily it. So I will highlight this as for you to remember. The beta tubulin sites are also overlapped. Okay. So beyond this, what else do we have is the structural features, which is comparing that why it is similar to Taxol, because it has the aromatic group for Taxol, similar aromatic group, which is a imidazole, like thioprazole. And then it has this phenylistosyrene side chain, whereas here, this side chain is existing as well. Instead of oxy-10 for Taxol, it has a hydroxy group. So these three features make it active and active in the same site as Taxol. That is primarily it. And the knowledge check, identify the pharmacophoric structures of all these, identify the targets for each of them, whether it's a microtubule polymerizer, polymerization stabilizer, or microtubule depolymerizer. That is all. And have a wonderful spring break. Those of you who are performing ROSA, all the best. Take care of yourself. Okay?
Apology for running over some time.
Yes.
Thank you for being patient. I don't want to